Welcome back, everybody. We're so glad to have you here for another day of learning. Up first, we're going to have Elle Geddes. She is a geneticist, a cardio geneticist out of Indiana. Unfortunately, she ran into a lot of travel issues and her flight was canceled and she was not able to get here in person, but she is joining us virtually. So we are excited to have you. Thanks, Elle.
Yeah, I truly am so disappointed I'm not with you guys right now. And I'm really, really honored that you guys have asked me to speak and share with you. And so today we're going to talk about the genetics of heterotaxy, and we're going to spend a lot of time sort of going over some of the general genetics and basics of genetics and genetic testing, because I think that background is really critical to understanding some of the more complex parts. And I think that aspect is also probably the most important to understand uh, as a parent clinically for patients with heterotaxy. And then we'll talk about a little bit about how left and right are established. And then clinically, what are the important aspects of genetics of heterotaxy and how do we think about it? And so I think of your body as being made up of the building blocks called cells. And in each cell, there is a library of information that makes up everything that's you, and that is the genome. And so I then divide that library into the bookcases, which are chromosomes. And you have 23 pairs of chromosomes. One comes from mom, one comes from dad. And on all the bookcases, we can look at generally what what are the number of bookcases visually, and what are there large missing or extra pieces. And so occasionally there can be flipped bookcases where a little piece of one chromosome is flipped onto the other, and those are called translocations. But generally, this test is not the most helpful for the average patient with heterotaxy and doesn't give us a lot of insight into why a patient might have heterotaxy. And then all the books are all your genes, and you can sort of read through them letter by letter. We sort of divide those books into so the way that we look at books most commonly is a chromosomal microarray or microarray or array. And that is sort of, are there missing or extra copies of the books or chunks of bookcases? And that looks just at ratios of DNA, which we call copy number. I am sorry, my mouse is a little too sensitive this morning. And so that has been classically the most common test we use to look for copy number. And until really the past few years, probably the main or most sensitive test we've really had routine access to. And so patients with heterotaxy should be assessed for if they have copy number variants or deletions or duplications or missing or extra pieces. But currently there are lots of other tests other than microarrays that can also give us an idea of copy number. So you don't necessarily have to have a microarray to tell if you have copy number variants. And so within those books, within the gene, there are different parts of it. And so if you think of a book, really the meat of it is the chapters and the text, but you know, the other aspects of the book are important and give context to, the cover and, the introduction, prologue, the dedication. Those are parts of the books that are important, but we don't really think about as much and don't focus on. And so we call those chapters, the meat of the book, exons. And then in between the chapters and the parts around the gene that are important to the gene, but less well understood and a little bit more subtle, we call those introns. And so mostly what we focus on are exons. And the reason these names are a little important is it sort of goes into the next parts that we look at. And so when we're reading through the chapters and really all of the gene, we look for sequence variants or typos. And there can be a whole lot of different typos. And so if you think of the line, look at all the bright colors, let's say that is the message of our gene. Well, you can have variations like colors spelled with a U that gets that message across, but is wrong according to spell check. And you can have even more obvious typos like spelling bright with a T, and that looks really wrong, but it still sort of gets the message across and works okay. You can have a much more subtle change, like changing the B or taking out the B, and it really disrupts the message, even though it seems like a sort of smooth line of text and that it could be right. You can have big disruptions, like totally taking out the word bright, but the message still kind of gets across. But if you do it in a different way, the message falls apart. And so that sort of leads into some of the complexity of when we're doing more specific genetic testing, because we have a lot to learn, and sometimes we mostly have the spell check. And so that leads us to the types of variants. And so you want to be really cautious about calling variants mutations or people describing variants to you as mutations, because that is a word that we've used for a long time, but don't like to use anymore because it can be really confusing in a setting where all of us have variants, and this testing is now sensitive enough to detect those. And the word mutation was supposed to be banished from medicine, which hasn't worked yet, in 2015. And that was really the first time the ACMG, or American College of Medical Genetics, released any formal guidelines as to how to interpret changes in the DNA or variants. And if you think about how long people have been working on genetics and doing genetic testing, 2015 isn't really that long ago, and it's still kind of subjective. And so we classify variants into 5 general types: benign variants, which are not disease causing; likely benign variants, which we think with a relative amount of certainty are not disease causing; and then variants of uncertain significance, which is the area where it makes the terminology kind of specific and important. And if you look at this graphic over here, the way that we classify variants, so this benign or not pathogenic, it's saying it's less than 0.1% of variant. The likely not pathogenic of little significance or likely benign is the 0.1% to 0.49 or less than 5% of variance. And then the variants of uncertain significance are 5% to less than 95% of variance. And so when you're actually looking at when we're doing sequencing and when we're doing genetic testing, 90% of the variants we come across are going to fall into the variant of uncertain significance category. So it's common, if not expected, when you do certain types of tests that you will get a variant of uncertain significance, at least one. And that's especially true in heterotaxy with ciliopathy genes. And then there is the likely pathogenic or likely disease-causing variants, which are, the top sort of 5% of variants. And then the definitely disease-causing or definitely pathogenic, the top 0.1% of variants. And that also sets us up for what sort of genetic tests we can do that look at variants. And so that generally falls into gene panels, exomes, or genomes. And that is why understanding, or at least having some awareness of the word exon can be important, because it's built into the testing name of some of these tests. And so gene panels, which until recently and in a lot of areas are still the only potential option for more advanced testing, the question we actually ask the test is, are there variants or typos in a list of genes? And so to answer that question, especially if we're looking at 100 or more genes, you would expect to get at least one variant of uncertain significance. So that test is likely to come back abnormal or with a mutation that might actually not be a diagnosis or be important to your child's health. And so that's where the terminology becomes important, because even healthcare professionals, even geneticists, will slip into old habits and call these mutations and say there's a mutation in this gene. And it's not too hard for that to sort of roll out into, well, they must have this diagnosis, when in reality, it's a variant of uncertain significance. It's possible that someday we will understand it to cause disease, but right now we don't think so. And so that's an area where the terminology becomes a little important and also really, really confusing. The test that we try to do is exome sequencing or whole exome sequencing. It's not actually whole. Every exome misses a few thousand genes, but they're typically genes we don't understand what their role is in disease. So we get a fairly good picture of what is going on. And as this little graphic shows, when we're talking about the exome or the exons, the sort of meat of the genes, it's only 1% of our whole library or whole genome. And that is the part of the library, the exome, that we understand the most, even though we still don't understand most of it. The other sort of nuance with this is that exome sequencing, when we run it, the question we're asking that test is, are there variants that explain what's going on with our patient that we're testing? And so that's an intrinsically different question than the question we ask the gene panel of, are there any variants in these genes? And so that's important for understanding that you are less likely to get variants of uncertain significance on exome. And if you previously had a gene panel that reported multiple variants of uncertain significance, they might not be reported in the exome. And that doesn't mean that the test didn't do as good a job. It just means they asked a different question. And it said, okay, those variants of uncertain significance, we don't think are important for this patient's health or what's going on with them. It's also important to know because if something new or different happens, like now we're having hearing loss, if that wasn't part of the first question, sometimes you need to redo the test and say, okay, we're going to reanalyze it, looking for variants that are associated with hearing loss. The caveat for heterotaxy is if heterotaxy is listed in the initial indication, even if we find new features of heterotaxy, the original testing should have reported it. So there's some sort of insurance, I guess, in using heterotaxy as a phenotype or term of why does this person have heterotaxy? The other test we hear a lot about is genome sequencing. Once again, it's not actually whole. It leaves out pretty large portions and chunks of the library. But once again, the parts that we are mainly interested in and focus on. And so that does most of our DNA and most of the book cases. And again, the question asked by this test is, are there variants that explain what's going on with our patient? And so some of the caveats to this testing is that it's highly, highly dependent on the details. So each panel or even each exome, depending on when and where it was run, may or may not include copy number analysis. So if you had an exome or a genome with copy number analysis, you don't need a microarray. That was looked at. And if it doesn't, you might also need a microarray to make sure we're looking at all the common causes we understand for heterotaxy. And as I mentioned, exome and genome are reported based on what we tell them about a patient. And so sometimes initially a patient will be identified to have one or two anomalies and the whole heterotaxy hasn't been put together. And in that case, those genes might not be primarily assessed or looked for, and that might be an indication for what we call reanalysis, or looking at all that data again and saying, okay, now that we're looking and focusing on genes associated with heterotaxy, do we find any changes that might explain what's going on with our patient? The other thing is I sort of alluded to in 2015, the American College of Medical Genetics made classification guidelines for exon or coding variants, but we have absolutely no guidelines on how to classify non-coding or intronic variants. So 99% of the library, we don't even really know how to interpret that into the set of 5 variant types. So to say we have a long way to go is an understatement. And so of our over 20,000 genes, over 20,000 books, we've associated disease with fewer than a third of them. So every year we add maybe 100 or 200 new disease associations, but once again, we have a long way to go. So when we do an exome or do a genome, really we're looking at less than a third of 1% of our DNA, just because there's so much left to learn about genetics, which also means that while genetic testing really feels like it should be a one and done, right? Like your DNA is your DNA, it can end up being a process over time because we get smarter, we get better technology, and it's something that you might need to update periodically. The other major caveat is that the way that we currently think about genetics is we think, okay, this gene does this thing. So this gene does your hair color, this gene does your eye color. There's sort of this straight association. And if a gene is broken or mutated or those sorts of things, then you have disease. But what really happens is there is a spider web of network genes together and signaling pathways. And so that is something that we don't really understand very well, other than we can often identify the pathways, but we don't know how to clinically say, well, there's a change in this gene and there's a change in this gene, and they work together, and therefore that means there's a problem. And I think that's really important in heterotaxy, because one of the things that we think about is cilia, which I'm going to talk about a little bit more later. And cilia are these big genetic machines that have lots of different proteins. And I think a lot of us believe that multiple variants in that machinery might cause disruption. And cilia are one of the first gene types where we actually found they were what we call digenic, where we actually are able to say, okay, if you do have a change in this gene and a change in this gene, it causes disease. And so they are one of the very first examples of how we're able to actually use that in clinic. And so I think that just adds fuel to our speculation that these variants are much more complex. And right now, on the clinical side, even on the research side, we don't have great ways to measure that overall pathway, how those things are interacting, and what might be a really complicated mechanism for disease. So to try to simplify that sort of, for lack of a better term, hot mess of connections and spider webs, I think of it as a game of telephone where one gene or one pathway is trying to send a message across. And you might have little inputs that sort of either disrupt the message or sometimes even correct the message and put it back on track. And so you might have something where each little change results in a disease versus that straight one broken gene equals one disease. So it might be that multigenic catalog. And the way that we describe that is the terms penetrance and expressivity. And so what this picture is a genetically purple bean and a genetically white bean are having babies. And the way that they have babies is actually all of these babies genetically should be purple, just like this parent bean. And so variable penetrance is what we say for, well, genetically they're purple, but they might or might not even look purple. And then when we say variable expressivity, we say, well, they're all genetically purple, but they might be different shades of purple. They might be light purple, they might be dark purple, everywhere in between. And the final way as well, there's variable penetrance and expressivity. So they're genetically purple. They may or may not look purple, and they might be all different shades of purple. And almost all of the genes that we understand for heterotaxy fall into this. And so that once again just adds further complications to interpreting the variants we get and determining the underlying cause of why someone has heterotaxy. And so these two terms together are the terms we use to try to explain, well, there's this spider web network that talks to each other and comes out on the other side, and maybe it's 100 different typos or variants, and maybe it's 5, but we don't really know. But that's why individuals with the exact same variant from the exact same family can have totally different types of presentations. And so that's really the sort of basic understanding of genetics and genetic testing. And once again, I've sort of alluded to it, but I also want to go into a little bit of the science of how when we're being created, left and right are established. And so first, don't start getting overwhelmed by this slide. I know there's a lot of pictures and terms, but generally when we're being created, we get 3 axes. We call it the anterior posterior axis, which is head to tail, the dorsoventral axis, which is back to belly, and then the left right axis, which is the one that we're most focused on. And so the left right axis is when we are able to say, okay, instead of being completely symmetric, let's give some differentiation to the organs in there. And one of the ways we do that is through ciliary function and messaging. And so to go into that a little bit more specifically, we have an organizational node that's responsible for signaling all of the left-right to the different parts that are ultimately going to form the organs. We call those the lateral plate mesoderm and ultimately the organ primordia. And so those terms aren't important. The concept of there is an organization structure that messages this is important. And the way that this does this relies heavily on cilia, or those little structures, and they both move the message. So they functionally will move the message, and they actually can sense the signal of, okay, I got this message. Now, what do I do? How do I form? How do I change this cell or this organization of cells? And so this picture shows, if we have normal ciliary movement, it doesn't account for ciliary signaling or sensing, then we can get sort of a straight message. And if we have disruption where maybe some of the cilia are turning backwards or not the way that we want, we can get disruption of that signal. And that can disrupt, well, this was supposed to be left and this was supposed to be right. And something that I think is important to understand, but also sometimes a frustration in the heterotaxy community, is that the heart is the very first organ that expresses left versus right. And it also ends up being one of the most sensitive to disruption of left versus right. So a lot of times heterotaxy is detected when we have a major cardiac anomaly. And particularly historically, but even still now, individuals who don't have a cardiac defect may not be clearly identified as heterotaxy and may not be, depending on the individual, in their interpretation of the literature, considered having heterotaxy. And so we're very cardiocentric, and myself included in heterotaxy, but it's mostly because of historically this, and still now, this is one of the first and most clear indicators of left-right axis disruption. And it's very frequently disrupted in patients with heterotaxy. But the more and more we understand, the more we understand that there are probably even more patients than we appreciate who genetically and embryologically have heterotaxy, but don't have heart defects and maybe aren't identified or put into this bucket. And that is really important because of a lot of reasons. One is identifying other systems involved, and the other is being able to identify if there are clear genetic ciliopathies can really change care, which I believe Dr. Saba is focusing on a little bit next. And so when we go into the genetics of heterotaxy, particularly the clinical genetics of heterotaxy as it stands right now, we can group the causes in multiple different ways. We can say, is heterotaxy the primary issue, or is it a symptom of a broader issue or broader genetic syndrome? And you might notice that I'm a stickler for terminology, but I think there is a method to my madness. And in this context, I typically don't use syndrome when describing heterotaxy, because the specific terminology of syndrome is supposed to say we have a clear genetic cause. And the genetic causes for heterotaxy are so broad and diverse. It can just be a symptom of something, or it can be the primary issue, that it's sort of hard to define it that way if you're being obsessive about the terminology. And so that's one reason why you might be like, why isn't she calling it heterotaxy syndrome? Why is she just saying heterotaxy? And it's because, well, this might be a symptom of something, or it might be the primary issue. And you guys might recognize this figure from the recent guidelines for clinical care that came out. But this is just a graphic of that. Is this associated with another syndrome or another genetic disorder that has a clear cause? And those can be chromosomal anomalies. Those can be, or complete bookcase differences. Those can be copy number variants or missing or extra books. And some examples of syndromes that can have heterotaxy as a symptom are things like trisomy 13 or 22q deletion syndrome. And then we have, well, single gene syndromes, or rarely digenic, of we know these two genes are disrupted. And primarily we think of primary ciliary dyskinesia. This example is Bardet-Biedl syndrome, which is a ciliopathy that focuses more on sensing cilia. And one of the disorders that we know, if you have this gene and this gene disrupted, you have disease. and generally ciliopathies overall, or it can be not associated or part of another syndrome. Heterotaxy can be the main focus, in which case we can have single gene etiologies or single book, and those can be lots of different inheritance patterns where of the two copies, you might only need one disrupted, you might need both disrupted. It might be linked to if you're a boy or a girl, and all sorts of things. Or this, what we're calling multifactorial digenic. Oligogenic is a fancy way for saying all those combinations of variants together, and plus or minus environmental factors, which might contribute to that game of telephone of disruption, or even sometimes correction. The way that I tend to look at heterotaxy is I think of primary patterning disruption. So, what is a disruption of the organizer's message, and ciliary dysfunction of how are these instructions being sent or sent. And when we look at the primary patterning thing, some example genes I think of are nodal, ZIG3, GDF1, FOXH1, ACVR2B. And so those names aren't important. You don't have to remember them. But even that isn't so clean. So for example, this ZIG3 gene, I classify it as a patterning gene. However, we think that it disrupts patterning by very early making the cell not able to put cilia in the right place. So could it also be in the ciliary dysfunction bucket? And then for cilia, we think of not just primary ciliary dyskinesias, though these example genes are mostly ciliary dyskinesias, but another one is Bardet-Biedl syndrome, which is a disorder that can cause short stature, it can cause obesity, it can cause polydactyly or having extra fingers, and but it's also, in an embryologic sense, a disruption of the cilia. And so when thinking about patients with heterotaxy, I think that all patients with heterotaxy deserve to be evaluated by a medical geneticist, because if there's one thing I think this talk has shown, it's that genetics is extremely confusing. But this is complicated by long wait lists and shortage of geneticists. For example, in the state I'm in of Indiana, we're over 27 geneticists short for taking care of the population. And so 27 geneticists when our group is 14 is a tall order to ask in expansion. And generally for each geneticist that's looking for a job or graduating, there are over 8 potential job openings for each one of them. And so that doesn't mean that patients with heterotaxy shouldn't see a geneticist. It just makes it a little bit more difficult. I think that ideally this should be done at birth, at least. And the reason for that is evaluating for signs of, is this a symptom of something, or is this the primary issue? And making sure that the right testing and comprehensive testing is sent as immediately as possible. So if there are clear answers we understand, we get that as soon as possible too. And I think patients with heterotaxy, their clinical genetic testing, when there's no clear known cause already, should at least include copy number assessment. Are there missing or extra books anywhere? And then gene sequencing or reading through letter by letter, at least focusing on primary ciliary dyskinesia genes, given how much that can change how we take care of it. And times to go back to genetics when you've done all the testing and there hasn't been a clear answer. Is something new or different is happening? Like the example I used of hearing loss. Reproductive questions or plans. You know, my youngest with heterotaxy is now 3. We're thinking about having another baby, or we're already pregnant. Can we update testing, see if there's anything we can better understand in regards to risk for future children, or test for, or prepare for? And then periodically for updates in knowledge and technology. And so we typically don't do this more often than yearly. Genetic knowledge moves incredibly quickly and also incredibly slowly. And so often for stable kids who aren't having major medical changes, I do it on more of a 3 to 5 year range to allow for significant advances and potentially significant new technologies or testing availabilities. And for more stable adults, sometimes more like 5 to 10 years might be appropriate. And so those are the things that I try to think of when, and I think are important for the clinical genetic assessment of patients with heterotaxy. The take-home messages are: genetics is complex and constantly changing. Your DNA doesn't change, but our ability to analyze or understand it will. It's all about the details. Abnormal testing or testing with a variant doesn't mean that you found an answer. And normal or non-diagnostic testing that didn't find a variant doesn't mean there isn't an answer. It's just not one we currently understand or can detect by the tests we did. And then the final one goes back to those purple beans. When a genetic cause of heterotaxy in a family is known, the severity of the laterality or the severity of heterotaxy can't really be predicted. We usually fall into that variable penetrance and expressivity. So is someone going to have a major cardiac defect? Is someone going to maybe have mostly a subtle interrupted inferior vena cava and bilateral right lung? Is someone all of the above? Sometimes is someone going to have this and not clearly have any laterality defects? It's all sort of unpredictable. And some of it is because of all the complex messaging and signaling that goes into establishing that left right. So even when you have a main driver, exactly how that message is moved by the cilia, exactly how that message is sensed, that can all be essentially somewhat random in that moment of creation or embryologic development. And we aren't able to detect that yet or predict that yet. And so with that, we have some time for questions. I know that's a lot of really complex material. So I'd love to hear what questions you guys have or what I could clarify better.
Hey, do we have any questions from people in the room or on the chat? You guys are welcome to weigh in through there. I'm going to hand it to Renee.
Hi. So I had a question of what happens. So my daughter had heterotaxy and holoprosencephaly, hemifacial chromoicrosomia, cleft lip cup palate, blah, blah, blah. So just a lot of stuff going on clinically, right? We did genetic testing, and ironically, she came back with a pathogenic variant for a disease she didn't have. Tuberous sclerosis, we'll say. It was an SE2 gene variant. And so like absolutely nobody's known kind of what to make of that as genetic testing. Like when you say like new technologies and new whatever, do those known kind of already pathogenic for something separate? Like how does that work? Does that like feed into information and then those things become reclassified? Does that make sense, that question?
Absolutely. Yeah. We frequently will find what we call incidental or diagnoses that when we do this broad testing that seem clear to be a genetic cause of that disorder, but may or may not be really associated with their symptoms. And so variant classification absolutely changes over time in all the ways, from disease causing to not disease causing, from not disease causing to disease causing, and from, thankfully, those ones are less common. And in the middle, those variants of uncertain significance up to disease causing down. And so that's another reason to periodically check in with your geneticist is, has this variant classification changed? Currently, that's a manual process. Can I check again to see what else is known about this variant? Has it been reported elsewhere? Those sorts of things. But sometimes, well, someday we hope that we'll be able to do like artificial intelligence type automated constant reclassification and then push out to individuals who have that variant and their medical teams of, hey, we reclassified this. The other thing is just over time, things and understanding changes. So geneticists have been wrong a lot. We've been wrong a lot. And for example, we've taken disorders and put them on newborn screening where every baby is tested for them on that heel prick shortly after birth. And then when we start testing whole populations, we figure out it's not even a disease. And so it's hard not to be humbled by being wrong so frequently. So I think that it's good to have a level of skepticism, especially of older reports. So anything that's over, 10 years old, that genetic testing and how it was interpreted, I'll often be skeptical and be like, I need to look at the data myself and see how I classify it. The other thing that's a little bit more complicated is that another part where geneticists have been wrong is due to the limited ability to do genetic testing, largely because of expense and partially due to technology. We've had this sort of self-fulfilling cycle of what textbook pictures of a disorder are. So if we have purple dot on your hand syndrome, similar to purple bean syndrome, and we say that, okay, everybody with this disorder has a purple dot on their hand. And so because testing is expensive and difficult, we only test people that have a purple dot on their hand. And so over time, it just sort of feeds on itself that everybody with this disorder has a purple dot on their hand, but also the only people tested have a purple dot on their hand. And so we're discovering now with broader sequencing that, hey, people that we thought had this disorder or would have never thought had this disorder actually do. And so every textbook definition of genetic disorders is sort of constantly expanding and becoming more fluid of what that can actually look like. And there's also the sort of difficulty of that happens as a geneticist. I see that weekly, if not multiple times a day, of this syndrome doesn't look exactly like it should. And especially for individuals with milder things, we don't publish that because the literature isn't really incentivized to talk about milder patients. Having an atypical presentation, we might be incentivized to publish, but not really milder things. And so even when you're trying to look up and find the textbook definition of a syndrome, it can be incomplete. And that's hard not only for geneticists, but particularly people outside genetics who don't see that every day, and other providers and caregivers who are like, that's not at all reported in this syndrome, that can't be related. Because it's almost stored as like a word of mouth sort of network between the geneticists or disease experts of, oh, XYZ always specializes in this, I'll ask them about it. And because everyone asks them about it, they tend to have a better idea of the spectrum. But once again, that's not really published anywhere or openly accessible. And so based on what you're describing, if there is an incidental diagnosis of tuberous sclerosis, I don't think that explains the symptoms. And not just because it's not textbook, because we are expanding those textbook definitions, but because at least for genetics, tuberous sclerosis is a more common disorder. But we have about 6% of the tests we run will have more than one genetic disorder detected. And the most I've personally diagnosed in an individual is 4 separate genetic disorders. I hope that actually answered your question.
I think it did. I'm getting a nod. Would you be able to look at, we've got a couple questions in the chat. Are you able to see those?
Yeah.
So first, are you someone who, so we have a family who's Zooming in. Their little one is actually admitted right now. They're going to see Dr. Fitzgerald Butt. And then with you, is Sarah going to do the same things at the first appointment that you would do?
No. So Sarah's one of our genetic counselors. I work with her very closely. She's part of our team. What we do typically at that first appointment is focus on the understanding of the genetic testing and what testing did we do, what variant did we find, that sort of thing, because especially in that whirlwind of neonatal follow-up, then it's hard to keep track of things. And all these people are coming to you with all these weird foreign terminology things, not even including genetics, let alone genetics. And the sort of take-home point might be that we didn't find anything clear on genetics, so you sort of forget about it. But what I like to do somewhat early is sit down and talk about, this is the test we did. This is what it can show us. This is what it didn't show us. These are those times you follow up. And then a little later, myself or Dr. Ware will check in and say, how medically are we doing? Do we need to do additional testing? Is there anything else we're considering? Though Sarah will flag to me like, oh, this came up at our appointment, can you take a look at it? And that sort of thing. So it's a little different focus, but part of the same team. And then And thank you for your kind words. That's very sweet. Thank you. And so when we're talking about being heterozygous or having one disrupted copy of a gene, which is really common in primary ciliary dyskinesia. So we have some sort of types of inheritance patterns. One we call autosomal dominant. I think of those as the Shakespeare books. Like that is hard enough to understand with baseline, let alone one copy's disrupted. Now I really don't know what's going on. And so for those disorders, just having disruption of one copy can result in disease. For autosomal recessive diseases, you need both copies disrupted. So I think of those as sort of like the C-Spot run book. It's like, well, I could kind of figure this out with only one copy. And so having one clear disruptive variant and disease-causing variant, but not having another one and having at least one copy that seems intact of that C-Spot run, then we would say, okay, they're a carrier. And so we would not expect them to have symptoms of that disorder. As I've sort of alluded to, it can be more complex than that, but that's why you can have a disease-causing variant, but not necessarily have disease, because you might have one working copy and that's all you need. And a lot of times in those situations, if we were to test the parents, one of them would have that exact same change. And that gives us further reassurance that that's not the main driver of what's going on in the child in front of us or patient in front of us. Any other questions or anything about those answers that didn't make sense?
Is it okay if I ask a question?
Go for it.
Is this one on? I don't think this one's on. Is that okay? Oh, it is. Okay.
I can hear you.
Okay. This is Dave Shidlow from cardiology at Children's. One of the things I and a lot of heterotaxy providers wonder about is the huge kind of range of things that we see in heterotaxy. For example, one individual with what at Boston Children's we would call polysplenia might just have interrupted IVC, and that seems to be it. And then another child might have interrupted IVC and PCD and biliary atresia and a hypoplastic left heart syndrome variant. And so what I wonder is where we are in terms of saying a genetic difference or a collection of genetic differences is going to be responsible for this manifestation or this set of manifestations in a particular patient.
Yeah, and so I think that there are a couple different levels and ways to think about that. So the first way I think about it is, well, what type of gene disruption is causing it, like patterning, ciliary, that sort of thing. But even in those moments of like, well, this is the pathway and this is all the different ways it can be disrupted, and it's this pathway and these are all the ways it can be disrupted, there is an element of randomness because it's, okay, we're pushing that signal protein over to where it will be sensed and detected. And so there are factors in there that are totally unpredictable because it's really in the moment, how fluid's flowing, how things are connecting. And especially if you seem to have a lower level ciliary disruption, I think that that can be really problematic and part of the complication for us figuring all of this out. Because when we try to figure out new ways of genetic assessment, we tend to focus on or try to focus on the most similar patients or the most severe patients, because in theory, they'll have the most obvious genetic difference. But in heterotaxy, I don't know that that's actually true genetically. And so there's that sort of random embryologic aspect. Another part that I think is interesting and evolving is, the machinery for cilia are used in microtubules and other areas of the cell that involve cellular migration and things like that. And so we know now that for cardiac development, cell migration is a major part of it. And one of the things that we've looked at previously is sort of burden of ciliary variants and in specific heart diseases and developmental differences, because neuronal migration is another big area of cell migration. And in those small core cohorts, the way that we've assessed it, we found, okay, the patients who have developmental delay are more likely to have way more ciliary variants than the patients with hypoplastic left heart or single ventricle heterotaxy who don't have developmental delay or don't have these features. And so Dr. Ware has a fairly large consortium of genome sequencing for patients with heterotaxy and are analyzing it in all those different ways. But one of the main complications, even in research, is how do you add up or combine genetic variants that are so specific and have different terminology, and like, how do you add those, or how do you rate those even in the same gene? And so some of the things that we've been experimenting with is there are prediction models that try to turn everything about that gene variant into a number. And it's like, okay, I can work with a number. It might not be perfect, but I can work with a number in figuring out number of variants and burden of variants. But then there's all these aspects of, well, each gene is a different length and might be more tolerant of disruptive variants or not. So this gene might have an average number in the general population of 16, and this gene might have an average difference in the population of 2. And so there's still a lot of problems of how do we normalize that and make that more concrete. And one of the ways people are doing things like that are polygenic risk scores, which is different than what we're talking about. But even those we've shown can, when applied clinically, can actually be detrimental to health, because essentially all of our genetic knowledge of what we use for detecting if there's a typo or not is based on individuals of European ancestry. So anytime we have anyone who has non-European ancestry, they're more likely to get rare variants or variants of uncertain significance just because they're not represented in our allele frequency databases, which I'm guessing you might be familiar with some of the large efforts to change that, because many of them are coming out of Boston and the Broad Institute. But when we use those polygenic risk scores clinically, we might cause more health disparities than already unfortunately exist. So there's the, how are we going to measure this? And then how are we going to, even for research, how are we going to then translate it clinically? And so once again, if we can work something into a score or number, that's something we can quantify clinically at least. But the ways to do that on things like breast cancer compared to something like heterotaxy, which is so much more rare, also adds a layer of complexity, because how do you verify it even if you get really good research data? And that's what the sort of collaborative Dr. Saba has set up and is leading, is really hoping to help us all work together and merge our data and try to strengthen it and have more of a think tank of how do we solve and tackle this problem. Once again, I'm not sure that answered your question. I hope it did.
Dr. Geddes, thank you. Tom Saba here. So so I had about 10 questions, but I can only choose one to ask right now. So I'm going to kind of use what you talked about a minute ago, because there was a lot in there. And I think it's important to understand that we just can't really predict all the medical issues of our patients based simply on their genetic tests. You know, I have a set of siblings, teenage boys I follow in my clinic, and they have the exact same genetic problem in a gene called CCDC39. And so they've lived their whole lives with this condition called primary ciliary dyskinesia. And one brother has heterotaxy and had lots of kidney issues and hearing loss and, well, multiple medical problems. And the other brother is really quite healthy and plays high school football. And I've never been able to really understand why they're so different if they have the same genetic abnormality. And so I'm guessing this is probably because there's much more to the pathogenesis of heterotaxy than just one gene, I think. So that's part one of my question. And part two is, if this is the case, if you agree that there's more than just one gene, then should we just go to whole exome sequencing on all of these patients rather than, you know, these commercial panels that look for single gene defects?
Yeah, so for part one, I think that, yeah, it's that multigenic sort of effect, but I think sometimes it's also just like medical natural history in some way of once you get, I think of sometimes our kids with genetics, and we'll see, and this isn't specific to heterotaxy, but we'll see kids with, for example, there's data to show that kids who undergo heart surgery who have a copy number variant have double the mortality of patients who don't have a copy number variant, all things else considered equal. But it can be any gene anywhere in the genome, and there's no clear cause of, there's no clear like it's mortality due to this, it's mortality due to this. It's just like, well, they're double the risk, and that's been shown at multiple institutions. So we know it's real, even though we have no understanding of why that would be the case. And so I tend to start to think of gene expression, because that's a whole nother piece of it, of like, okay, this library in my skin is the same as in my heart, but they're totally different. And so the gene expression part is modifiable and heritable in some ways. And I think that when there's imbalances or disruptions, you can get cellular energy dysfunction, like you're making the same protein over and over again, which maybe you don't need, but you can't turn off the signal because that protein doesn't work, or that protein is disrupted and not turning off the signal. So your cell's doing a bunch of stuff it doesn't need to do and wasting energy. And I think of it as sort of overall medical reserve of, if I'm spending a lot of time doing stuff that really doesn't result in anything helpful, even a little time doing stuff that's not necessarily helpful, I have less reserve for when something happens that I need to be more efficient and I need to fix. And so I think sometimes it just sort of snowballs out of control of, I had decreased reserve and this one thing happened, and now it's just sort of each additional thing adds on, and it's because I can't, like my bucket's already full. It doesn't take much to make it overflowing. And once it's overflowing, sort of stopping that takes more for me. And I think that is very true in our heterotaxy patients, partially because of the multi-system involvement of it can tip off lots of different things. But I think there is some, maybe he has more problems because his reserve is less than his sibling who doesn't have these congenital anomalies, in addition to potentially having those different modifying changes in various pathways. And I think that in an ideal world, every patient with heterotaxy should have rapid genome sequencing at birth, but what we can get insurance to pay for, and as we get more complex testing, what we can get people to support us explaining it and interpreting it becomes more problematic. So I would absolutely love to and prefer to do exome testing over panels. And I'd love to do genome testing over exome testing. And part of that is, yeah, I know the genome, most of which we can't interpret right now, but that sort of longevity of 5 years from now, I can reinterpret it and it'll be better versus 5 years from now, instead of reinterpreting it, I'm going to do a genome then. I'm not sure that answered your question either, but you know how to get a hold of me with the 10 other questions too. So, and everybody can get a hold of me. My email address is on the first slide.
I think that is all the time we have for this. I know we all have 10 or 15 or 20 other questions, so we'll refer you back to the first slide. You can send them straight to Dr. Geddes' email. Thank you so much. This was incredible, and we have so much to think about.